And if beauty is terror, said Julian, then what is desire? We think we have many desires, but in fact we have only one. What is it?
Hi everyone, I'm Skye, and tonight I felt like a change of scenery, so I came to a coffee shop, grabbed a drink, and thought we could study together. If you're looking for a simpler way to learn medical and science topics, you're in the right place. So while we're here, let's talk about meiosis. Let's start with the big picture. Meiosis is a special type of cell division that produces gametes or sperm and egg cells. These are the cells needed for sexual reproduction. And yes, meiosis doesn't just happen in humans, it also occurs in plants, fungi, and many other organisms. But to keep this lesson focused and easy to follow, we'll be looking at how meiosis works in humans. So for now, let's stick with humans. Unlike mitosis, where one cell divides into two identical cells, in meiosis, it divides into 4 cells. Each of those cells has only half the usual number of chromosomes. That's important because when a sperm and egg come together, the baby gets the correct number of chromosomes. Meiosis also creates genetic diversity, so each sperm or egg is a little different. That's one reason why every person is unique. Now that we know what meiosis is, let's discuss why the chromosome number has to be cut in half. The main purpose of meiosis is to reduce the chromosome number from 46 to 23. As a result, it produces haploid gametes, meaning sperm and egg cells each carry only one set of chromosomes. During fertilization, the sperm's 23 chromosomes combined with the egg's 23 chromosomes, bringing the total back to 46 chromosomes. Here's a simple analogy. Imagine a treasure map that's split into 2 halves. One person has one half and another has the other. In the same way, the sperm and egg each carry half of the chromosomes needed to form a complete set. Before we continue, here's a quick reminder. Sometimes errors can happen during meiosis, leading to conditions such as Down syndrome. Likewise, topics like how identical twins form involve additional concepts beyond the normal process of meiosis. In this video, we're only discussing about the normal process. Moving on, let's take a look at a few important terms that will come out throughout this lesson. A diploid cell contains 2 complete sets of chromosomes. In humans, most body cells are diploid, which means they have 46 chromosomes. A haploid cell, on the other hand, contains just one set of chromosomes, or 23 chromosomes in humans. During meiosis, matching chromosomes pair up in a process called synapsis. Once they're paired, they form a structure known as a tetrad. A simple way to think about this is matching socks. Each sock finds its matching pair, just like homologous chromosomes pair up during synapsis. Now that we've covered these key terms, let's look at one of the most important events in meiosis. One of the most exciting events happens after these chromosomes pair up. They exchange small pieces of DNA in a process called crossing over. This happens during prophase 1 at points called chiasmata. Because the chromosome exchange genetic material, each gamete ends up with a unique combination of genes. That's one of the reasons brothers and sisters can look alike but still have many differences. Here's a simple analogy. Imagine 2 friends trading stickers. After they trade, both collections are a little different than before. Crossing over works in a similar way because the chromosomes exchange pieces of DNA. Before meiosis officially begins, though, the cell still has one more thing to do. It needs to prepare. Before meiosis begins, the cell goes through a preparation called interphase. During this stage, the DNA is copied so that each chromosome has an identical chromatid. The cell also grows and gets everything ready for the 2 rounds of cell division. You can think of like making a photocopy of your notes before sharing them with your classmates. You want to make sure everyone receives a complete copy. Now that the cell is fully prepared, meiosis can finally begin. The first round of cell division is called meiosis 1. It's also known as the reduction division because this is where the chromosome number is reduced from diploid to haploid. During this stage, the homologous chromosomes separate while the sister chromatids stay attached. Imagine two dance partners walking to opposite sides of the room, but each person keeps their own backpack. The partners separate, but they don't leave their belongings behind. In the same way, the homologous chromosomes separate while the sister chromatids remain together. The first and one of the most important stages of meiosis 1 is prophase 1. Now we've reached prophase 1, the longest and one of the most important stages of meiosis. During this stage, homologous chromosomes pair up through synapses, forming tetrads. They also exchange pieces of DNA through crossing over, creating new genetic combinations. Many of the events that make meiosis different from mitosis happen during this stage. To picture this, imagine two friends exchanging friendship bracelets. After they swap them, each person has something new from the other. Crossing over works in a similar way because chromosomes exchange small pieces of DNA. Next, we'll take a closer look at the 5 substages of prophase 1 and see what happens in each one. As we mentioned earlier, prophase 1 is such an important stage that is divided into 5 substages. A simple way to remember them is with the mnemonic LZPDD. First is leptotene, where the chromosomes begin to condense and become visible. Next is zygotene, where homologous chromosomes pair up through synapses. Then comes pachythene, where crossing over takes place. After that is diplotene, where the homologous chromosomes begin to separate but remain connected at the chiasmata. Finally, diakinesis is the last preparation stage before the chromosomes move to the center of the cell. You can think of it like getting ready for a school presentation. First, you organize your materials, then meet with your partner, exchange ideas, prepare everything, and finally walk to the front of the classroom. Each step gets you ready for the next, just like the substages of prophase 1. Now that prophase 1 is complete, the chromosomes are ready to line up in the center of the cell. During this stage, the paired homologous chromosomes, also called tetrads, line up in the center of the cell at the metaphase plate. At the same time, spindle fibers attach to each homologous chromosome from opposite poles. These spindle fibers will later help pull the chromosomes apart. Imagine students standing in pairs on the center line before a relay race begins. Everyone gets into position before moving in opposite directions. Now everything is in place and the chromosomes are ready to separate. Now let's move on to anaphase 1. During this stage, the homologous chromosomes separate and move toward opposite poles of the cell. One important thing to remember is that the sister chromatids stay attached. Only the homologous chromosome pairs separate during meiosis 1. A simple way to picture this is to imagine two teammates heading to different classrooms while each keeps their own backpack. They separate, but they don't leave their belongings behind. In the same way, homologous chromosomes separate while sister chromatids remain together. Once the chromosomes reach opposite sides of the cell, it's time for the cells to divide. During telophase 1, the chromosomes reach opposite poles of the cell. Next is cytokinesis, where the cytoplasm divides, producing 2 haploid daughter cells. Even though the chromosome number has already been reduced, each chromosome still consists of 2 sister chromatids. Imagine using a divider to split one large classroom into 2 smaller classrooms. The students are now in separate rooms, but each still has all of their belongings. That's similar to what happens during the stage. Before the second division begins, the cell goes through a short resting stage. This short resting stage is called interkinesis. It takes place between meiosis 1 and meiosis 2, giving the cells a brief moment to prepare for the next division. Unlike interphase, the DNA is not copied again because it has already been replicated before meiosis 1. Think of it like taking a short break after finishing one exam before starting the next. You take a moment to prepare, but you don't go back and repeat the exam you've already completed. After this short pause, the second round of meiosis begins. The second round of cell division is called meiosis 2. It's similar to mitosis. The main difference is that it starts with the two haploid cells produced during meiosis 1 instead of diploid cells. The main purpose of meiosis 2 is to separate the sister chromatids. A simple way to picture this is by unzipping a jacket. At first, both sides stay connected, but once you pull the zipper down, they separate completely. That's similar to what happens to the sister chromatids during this stage. Now let's see how this happens in each stage of meiosis 2. Meiosis 2 follows the same 4 stages as mitosis. During prophase 2, the chromosomes condense and spindle fibers begin to form. In metaphase 2, the chromosomes line up in the center of the cell. During anaphase 2, the sister chromatids finally separate. Finally, in telophase 2, new nuclei form, cytokinesis takes place, and 4 genetically unique haploid cells are produced. Here's one last analogy. Imagine 4 balloons tied together with strings. Once the strings are cut, each balloon becomes separate on its own. In a similar way, each daughter cell becomes its own individual cell at the end of meiosis 2. Now that we've gone through both rounds of cell division, let's look at the final result. At the end of meiosis, 4 haploid daughter cells are produced. In humans, each cell contains 23 chromosomes. These cells are also genetically unique because of crossing over and the random separation of chromosomes during meiosis. Over time, these cells will develop into sperm cells or egg cells, making sexual reproduction possible. Here's a simple way to picture it. Imagine a baker using one batch of dough to make 4 cookies, then decorating each one differently. They all came from the same dough, but each cookie is unique, just like the 4 daughter cells produced by meiosis. Now that we've seen the final result, let's compare meiosis with mitosis. Although mitosis and meiosis are both types of cell division, they have different purposes. Mitosis produces 2 genetically identical diploid cells and is mainly used for growth, tissue repair, and replacing damaged cells. Meiosis, on the other hand, produces 4 genetically unique haploid cells that eventually become sperm or egg cells for sexual reproduction. Another important difference is that crossing over and synapses happen only during meiosis 1. They don't occur during mitosis. Think of it this way. Making photocopies gives you identical copies every time. That's like mitosis. Now imagine baking 4 cupcakes and decorating each one differently. They all came from the same batter, but each one looks a little different. That's a lot like meiosis. Now that we understand the difference, let's look at what happens after meiosis. After meiosis, the body goes through a process called gametogenesis, which is the formation of mature reproductive cells. There are 2 types of gametogenesis. Spermatogenesis produces sperm cells in the testes, while oogenesis produces egg cells in the ovaries. Although both processes involve meiosis, they produce different types of reproductive cells. You can think of it like a factory making different products using similar machines. In the same way, meiosis is involved in producing both sperm and egg cells. But the final products have different structures and functions. Next, let's take a closer look at oogenesis. During oogenesis, only one functional egg is produced. The other smaller cells are called polar bodies. They usually degenerate because they receive very little cytoplasm. Most of the cytoplasm, organelles, and nutrients stay inside the egg cell. These materials support the embryo during the early stages of development after fertilization. Here's an easy way to picture it. Imagine 4 siblings sharing supplies for an important project. One sibling receives most of the materials while the other receives very little. In the same way, the egg cell keeps most of the nutrients needed to support early development. Before we finish, let's do a quick recap of what we've learned today. Let's quickly review the main points. Meiosis is a special type of cell division that produces 4 genetically unique haploid cells. It happens in 2 rounds of cell division called meiosis 1 and meiosis 2. One of the key events is crossing over, which creates genetic variation and helps make every individual unique. Most importantly, meiosis keeps the chromosome number the same from one generation to the next, making sexual reproduction possible. To wrap things up, imagine a family recipe being passed down through generations. Each person follows the same recipe but adds their own small touch. The recipe stays the same at its core, yet every version is a little unique. That's similar to how meiosis passes on genetic information while creating new combinations. And that's everything for tonight's lesson on meiosis. I hope the explanation made the topic easy to understand. If my voice sounds a bit different, I apologize. I'm a little under the weather. Thank you so much for studying with me. If this video helped you understand meiosis, don't forget to like, subscribe, and turn on notification bell so you won't miss future medical or science videos. Until next time, study smart, stay curious, and I'll see you in the next one.